Hi everyone, welcome back to Useful Genetics. This is lecture 5K, where we're going to continue our discussion of the two-hit model of how cancers get started, now applying it to the more complicated situation of BRCA genes and breast cancer. We'll talk about the two BRCA genes, BRCA1 and BRCA2. They work together in a complex, and so the consequences of mutations are very similar. We'll talk about whether cancer risk of this type can be said to be inherited dominantly, and we'll consider the roles of other genes in breast cancer. So it's very well established that mutations in the genes BRCA1 and BRCA2 greatly increase a woman's risk of getting breast cancer. They also increase a man's risk of getting breast cancer if he should inherit two defective alleles. They're both important in a particular kind of DNA repair. Remember DNA repair from Module 2. BRCA1 and BRCA2 proteins carry out a kind of DNA repair called homology-directed repair of DNA breaks. This is extremely important in maintaining the genetic stability of chromosomes, because otherwise broken chromosomes often don't get repaired. They're both said to be part of what's called a genome surveillance complex, a large complex of proteins that interact to look after the integrity of the genome, to make sure things don't go wrong, and then to prevent cell division when things do go wrong. So women who inherit one BRCA minus allele, either BRCA1 minus or BRCA2 minus, have a lifetime risk of breast cancer of 50 to 70%. And they also have a high risk of ovarian cancer. Women who inherit normal alleles have a risk of breast cancer that's maybe 8 to 10%, depending on genetic background and many other factors. This is a diagram of known mutations in, now I can't remember if this is BRCA1 or BRCA2, but you can see there's a wide diversity of mutations, nonsense and frameshift mutations that terminate the protein, missense mutations that change an amino acid, splice site mutations that prevent normal splicing of the messenger RNA. All of these are mutations that eliminate the function. You'll see that there's only a small number of missense mutations because only very particular single amino acid changes are going to ruin the protein completely, but that many mutations that frameshift the protein destroy its function. Now, the two-hit model of cancer development explains why inheriting a single BRCA minus allele causes a high risk of breast cancer. Because a cell will inherit, a person will genetically inherit one defective allele. So they'll have a genotype that's plus minus one functional allele, one defective allele in all the cells of their body. But only cells that undergo a second mutation to destroy the other BRCA allele of the same complex, of the same gene, will go on to develop cancer. But because this could happen in any cells in the breast, the risk is quite high that at least one breast cell will suffer the second mutation that creates a person with no BRCA1 or no BRCA2 function. But it's quite complicated. Unlike a retinoblastoma, where it's clear that the single mutation causes cancer, for BRCA mutations, it's more like the mutations set the stage for cancer because cells with two defective BRCA alleles, it's not that they're all set to divide when they shouldn't, but they go on to accumulate many other mutations. Because their DNA repair is defective, the BRCA mutations just sort of set the stage for all the other mutations that are then going to cause aberrant cell growth. And it then becomes very difficult for researchers to track the actual causes of tumor behavior because these cells have accumulated so many mutations. And you'll see this in a few minutes. Now, people often say that BRCA1 and BRCA2 are dominant genes causing high risk of breast cancer. And they say the same thing about retinoblastoma, that it's a dominant cancer risk gene. But going back to our previous discussion of dominance in Module 3, I want to pull that idea apart. So we know that individuals with 2 normal alleles have normal risk, and that individuals with 1 mutant allele have high risk. But before we can really say that the risk is dominant, we would need to know the phenotype of the individuals with 2 defective alleles. And that's a bit of a problem, because in fact, no person has ever been discovered who has 2 defective alleles. It's thought that having 2 defective alleles of the BRCA, either BRCA1 or BRCA2, is a lethal birth defect. And the same is true for mutations in RB. No individuals are known who have survived with no functional RB allele. And I showed you similar data for RB. Here's the interaction data for BRCA1. This is all of the genes that the BRCA1 protein interacts with. You can see then that this is an extremely important protein for all the cells in our body, interacting with many different other proteins in many different cell types. Here's a diagram showing the different parts of the BRCA1 gene. So here's the BRCA1 protein represented linearly. And then here are the different parts of the protein that interact with other different proteins. Again, this is giving you the sense of how all of our genetic functions are interacting with each other in this integrated, very complex genetic system. So this one protein, this one DNA repair protein, is interacting with all of these other proteins, including, there's our friend retinoblastoma. All of these are interacting with BRCA. c-MYC is a cell growth gene. RAD50 functions in mitosis. I don't actually know. There's BRCA2. Large numbers of interactions. Now, 90% of breast cancers arise in women who have normal BRCA alleles. Most breast cancer is not caused by mutations in BRCA1 or BRCA2. So what causes these mutations? This is really the really big issue for understanding and preventing breast cancer. And the unfortunate answer is we really don't know what causes breast cancer. There's lots of genes involved. There are lots of genome sequencing studies being carried out to identify candidate genes, as well as genome-wide association studies to look for other risk factor genes. Here in particular was a set of 5 papers published together in the journal Nature, a couple of years ago. All of these were genomic analyses of breast tumors, and 2 of them were published open access so that anybody can read these articles for free. The other 3 articles you'll have to pay for, but these 2 are open, and I've put links to them on the Module 5 module page in the suggested recommended reading. You certainly don't have to look at these. These are technical articles. They're quite difficult. But if you're really interested in the origins of breast cancer, you might want to look at them. Or there's an associated article, what's called a News and Views, that summarizes the results of all 5 research papers. So I'm only going to show you a little bit of data from one of these papers. But before you can understand the data what I'm going to show you, I have to introduce a new kind of genome analysis plot. You'll remember the Manhattan plots that we looked at for genome-wide association studies. What I'm going to show you next are Surcos plots, and these are plots of the genetic properties of one, in this cases, it'll be of one tumor. And just like the Manhattan plot arrayed the chromosomes in a line along the x-axis of the graph, here the chromosomes are arrayed in a circle. So here's chromosome 1, chromosome 2, 3, chromosome 4, chromosome 5, chromosome 6, all the way around. There's the X chromosome and the Y chromosome in a circle. The mutated genes that were found when this tumor was sequenced are indicated by writing the gene names at the location of the genes on the chromosome. Now, you can't read these gene names because of the poor resolution of this figure. Changes in the structure of the chromosome, small insertions and deletions, or chromosome rearrangements within a single chromosome are indicated by blue lines on the inside of the circle. And rearrangements between different chromosomes, where chromosomes are broken and rejoined to parts of other chromosomes, are indicated by these swooping green lines. You'll learn more about different kinds of rearrangements in Module 10. Okay, now we're going to go on and we're going to look at 6 Surcos plots for 6 different breast cancer tumors. And here they are. There's 3, this is data from a much larger set of, I think, 100 tumors that they sequenced. Here are 3 of the kind of breast tumors that are described as being estrogen sensitive. Most breast tissue cells are sensitive to the hormone estrogen, and cells will divide in the presence of estrogen. These breast tumor cells will divide in the presence of estrogen, but will not divide if the patient is given an inhibitor that prevents production of estrogen. So these cancers are easier to treat. These cancers are insensitive to inhibitors of estrogen production. They don't care about estrogen. And one thing you'll notice very quickly is that there are many more mutations in the estrogen-sensitive tumors, many more mutations all the way around, and many more rearrangements than in the estrogen-sensitive tumors. It's not clear at this point what's cause and what's effect, whether the mutations, these were tumors that happened to get a lot more mutations, and one of those mutations made them estrogen insensitive, or whether somehow being estrogen insensitive then causes the tumor to accumulate more mutations. But this gives you an idea of the genetic complexity of breast cancer tumors and of the diversity of these tumors. Each of these tumors is completely different. Well, no, that's not quite fair. They're not completely different. There are mutations that were found, what are described as recurrent mutations, that is, mutations that were present in more than one of the tumors that was analyzed. So in this study, here's the actual numbers. They sequenced the whole genomes of 46 tumors, and they sequenced the exomes of 31 more tumors. And of course, they had to sequence the normal tissues of these patients as well for comparison. And these are genes where mutations appeared more than once. They were in kinases that are involved in controlling cell division. There's retinoblastoma, a large number of different genes. And these genes are important because genes that are mutated in more than one cancer, more than one tumor, could be indicating that these are genes that are particularly likely to play a role in causing tumor growth. And so one of the things that the authors did in this study was they did a statistical analysis of how unexpected was it that you'd see 13 mutations in the same gene in, 77 tumors that 13 of them would have mutations in this gene, or that 4 of them would have mutations in this gene. And these are all what they considered to be statistically significant occurrences of more than a single mutation in a gene, indicating that there's probably a causal effect going on here. Now, the next graph just shows a couple of the genes that were analyzed. In this case, they looked at a total of 319 tumors, and they found 10 mutations in one gene called MAP24K. That's a regulatory gene, regulates cell division, and there's all the different mutations. They found 49 distinct mutations in another kinase that regulates cell division. In fact, they found 50 mutations, because in one case they found exactly the same mutation twice in 2 independent tumors. Again, this is indicating genes where mutations may actually be causing the tumor behavior. So what we've done, we've extended the simple 2-hit model for cancer that we talked about for retinoblastoma to the more complicated situation of the BRCA1 and BRCA2 oncogenes. We've examined other causes of breast cancer. We've looked at all of the different mutations that breast cancer tumors can have, and gives us a sense of how big a problem it is to identify the commonalities in breast cancers that will allow more efficient treatment. And we talked about a particular clue to commonalities, which is the presence of recurrent mutations found in multiple independent tumors. Now, coming up next, we're going to consider 2 more kinds of cancer, prostate cancer and colon cancer. Again, thinking about the different kinds of causes of cancer risk. I hope to see you there.